Welcome to the problem-solving set on the trip operon attenuation. Question 1. In a repressible operon, the molecule of focus is often the product or substrate in the pathway being regulated. It binds to the co-repressor or repressor, rendering it active or inactive. So we're going to go ahead and pick the correct term for each of these blanks. So a repressible operon, for example, tryptophan, which is the operon of focus for this chapter, the molecule of focus is often the product of the pathway, right? So in the lac operon, the focus was on the substrate, which was lactose, which would then be degraded. But here, the focus is on the product. So for the trip operon, the structural genes form the pathway that lead to the biosynthesis of tryptophan. So it's the product of that pathway. And the molecule of focus, tryptophan, binds to the repressor. So there's already a repressor protein in the cell, and tryptophan here, in this case, is equal to the co-repressor. And when it binds to the repressor protein, it renders it active. Okay? So remember, we're talking about a repressible operon, which means it's normally on, but if you have the co-repressor, tryptophan, that activates the repressor by binding to it, and that makes the repressor active, so now the operon is turned off. So in this case with tryptophan, a cell will continually make tryptophan. It will have their trip operon be expressed. But if there's enough tryptophan in the cell from outside, or they just made enough, then the excess tryptophan will bind to the repressor protein, which will then bind to the operon and stop transcription. Problem 2. The first domain of the trip leader sequence encodes for a short peptide with tryptophan residues. Explain the importance of this as it relates to attenuation and the regulation of the trip operon. So I've drawn a trip L, which is the leader sequence, and remember that it goes from 5' to 3', and these 4 domains in the leader sequence come before the structural genes of the trip operon. Okay, so what this is, it's a small piece of DNA that when transcribed into mRNA can form 2 different secondary structures, and the way that these secondary structure forms are regulated by how much tryptophan is in the cell, and therefore can regulate if the trip operon stays on or is turned off. So the secondary structure that's going to form is called a stem loop structure or a hairpin structure, and it looks something like this. And this is where you have bases of the mRNA base pairing together to form this secondary structure, stem loop or hairpin. Now this secondary structure, remember this is forming from the mRNA, and it can form between either regions 2 and 3 or between regions 3 and 4. And how much tryptophan is actually in the cell is going to determine which of these pairs are going to stem loop together. So how does the cell do this? How does it know which regions to go together? So as it says in the problem, the first domain is going to make a short peptide. So I'm going to draw that as just a few circles for amino acids. And it does have a few tryptophan residues in it, so I'm going to draw the tryptophan in red. Now remember that this short peptide, first the DNA, which is what's written, has to be transcribed into mRNA, and then the mRNA has to be translated into a peptide. So I'm going to write translation here. And to go from mRNA to amino acid, you need a translator, right? So this is what the ribosome's function is, and also the function of tRNA. So you need a ribosome, now remember that each amino acid has its own tRNA, which has a specific sequence that reads the codon, and brings over the correct amino acid. So what we need specifically for this is we need tRNA charged with a tryptophan residue. So that way, these tryptophans in the small protein from region 1 can be incorporated into the polypeptide chain. So tRNA for tryptophan is only charged with tryptophan if there's an excess of it in the cell. So think about it. If there's no tryptophan in the cell at all, these tRNAs aren't going to be able to be charged with any tryptophan because there's none around. So this is how, this is what attenuation is, and this is how it kind of reads how much tryptophan a cell actually has. So let's think about that. So let's look first at low tryptophan levels. If there's low tryptophan, that means that there's going to be low levels of tRNA charged with tryptophan. So when region 1, the mRNA from region 1 is going to be translated via a ribosome, the ribosome is going to stall on that mRNA because it can't bring over any tryptophan because there's no tRNA carrying tryptophan. So the ribosome will stall on region 1. And because it's stalled, regions 2, 3, and 4, the mRNA for these regions, are free to form structures with each other, and 2 is free to form structure with 3. So when there's low tryptophan levels, let me put tryptophan. When there's low tryptophan levels, the ribosome stalls on region 1, and regions 2 and 3 make this stem loop structure together. Now, when regions 2 and 3 stem loop together, this is where the operon will continue to be transcribed. So transcription continues. Which, in other words, means that the operon is on. And that makes sense, right? If you don't have any tryptophan, you want the operon that synthesizes tryptophan to be on and expressing its structural gene so that the biosynthetic pathway can continue. Now, if tryptophan levels are high, translation of region 1 is going to be fast, right? Because the ribosome won't stall on these tryptophan residues because there'll be plenty of tRNA with tryptophan around. So translation is quick. And because translation is quick, now remember that transcription and translation in a prokaryotic cell, like a bacteria, can occur basically together. So right after transcription happens, a ribosome can come to the mRNA and start translating it. So because it's so quick, region 2 now is going to be not fully incorporated into the ribosome, but it will be complex with the ribosome so that it's not free to bind with region 3. So region 2 is complex with ribosome. And therefore, region 3 and 4 now form this stem loop structure. Now, when this hairpin forms, this actually stops transcription, which is attenuation. Okay, and the reasons why these different structures stop translation or let it continue is really a biochemical topic, and you don't really need to know the technicalities of it. You just need to know that if 2 and 3 are stem looped together, these regions, transcription continues and the operon is on. But if regions 3 and 4 form a stem loop, transcription stops, and that means that the operon is now off. And that also makes sense if you think about it. If there's a lot of tryptophan in the cell, enough that there's plenty of tRNA charged with tryptophan, and the translation of region 1 can continue quickly, then there's no real reason to have the operon on to synthesize more tryptophan because you have enough of it. Okay? So just to recap, attenuation, which is another level of control. So you have the repressible control with the repressor, co-repressor, but then you also have attenuation. And this, the definition of it is the slowing of translation of the leader peptide, which is really the peptide that comes from the translation of region 1. So I'll just put region 1. And the slowing of translation leads to the formation of the 3,4 hairpin, which terminates the message or transcription. Okay, so this is just another level of control. It's what makes the trip operon very unique, that it has this attenuation control. So again, to recap, in order to have charged tRNA with tryptophan residues, there has to be plenty of tryptophan in the cell. So when tryptophan's low, that means that tRNA tryptophan is low, and the ribosome is going to stall when translating region 1, and therefore hairpin loop 2 and 3 will form, translation continues. When tryptophan is high, that means that tRNA tryptophan is high. Translation of region 1 is quick, and it's so quick that domain 2 gets sort of complexed into the ribosome, can no longer bind with region 3, so region 3 and 4 form the stem loop, and that stops transcription. So when you have enough tryptophan, you don't need to waste energy making the enzymes to make more of it. Problem 3. Any gene in the TRP operon can undergo mutations that lead to a loss of function. Predict the phenotype of the following mutants, choosing from the 4 choices below. Consider repressible regulation only, not attenuation. So before we go ahead and answer these questions, I want to go over the tryptophan repression. So you have a repressor molecule, which we'll call the gene for it TRPR, and this is always expressed, and this leads to the repressor protein. But in the case of the TRP operon, this repressor protein is made inactive. So it's always expressed, but it's inactive in its natural state. So for the TRP operon, this operon is always on until there is enough tryptophan in the cell to turn it off. Okay, because tryptophan is one of the 20 amino acids. It's required for the cell to do various cellular activities to make its amino acids, its proteins. So you always want to have this operon on until you don't need it anymore. So this repressor protein is made inactive in its normal state. When there's excess tryptophan, the tryptophan will bind with the repressor protein, making it active. So I'll call that the TRP plus R for repressor complex, can then bind to the operator of the TRP operon and turn the operon off. Okay, so the repressor protein's in its inactive state and it's in natural, but then when there's an excess of tryptophan, the tryptophan can bind to this repressor protein, making it active, and then the complex of tryptophan, which is the co-repressor, and the repressor protein is now active, can bind to the operator to block transcription of the operon. Okay, so now let's look at part A. The mutant that we have is a TRPR mutant, in which the repressor protein cannot bind tryptophan. So what did we just go over? That the tryptophan repressor protein is normally inactive, right, until it binds tryptophan. But in this case, this repressor protein can never bind tryptophan, and therefore the repressor will always stay inactive. So if it's always in its inactive form, that means it can never bind the operator to stop transcription, and therefore the TRP operon will always be on. Because if we're not considering attenuation here, we're just talking about the repressible regulation. If the repressor can never bind the operator, then the operon will always be on. So the phenotype here would be choice number 3, where the TRP operon will be constitutively expressed, which means always. So repressor will be inactive, never bind operon. Operator. Okay. B. A merodiploid in which a wild-type copy of the TRPR gene is introduced into the TRPR mutant. So remember, a merodiploid is where you have a cell, and some of its genes, not the entire genome, but some of its genes are duplicated via a plasmid. So a plasmid in this case, will be introduced into the cell that has a wild-type copy of this repressor protein. So now you're making a repressor protein that can bind tryptophan to become active and block transcription. And remember, because a repressor is a protein, it is a trans-acting element, which means that as long as there's one wild-type copy of this gene, even though it's from the plasmid, the protein can go over and work on the genomic copy of the operon. So because it's a trans-acting element, and now you have a functional copy of the protein, it can respond to tryptophan levels just like a wild-type would, and therefore it will restore the natural phenotype, the normal regulation of the operon. So for this, the normal regulation of the TRP operon is actually choice 2, and that's where the TRP operon is expressed only in the absence of tryptophan, which remember is opposite from the lac operon, which is only expressed in the presence of lactose. Okay, so you have a transacting element reintroducing a wild-type copy of this repressor protein can bring you back to the normal phenotype of the TRP operon, which is number 2. So let me just make a note of that. That is wild-type phenotype. Okay. Part C. A mutant in which the operator cannot bind the repressor. So in this case, they really mean the repressor-co-repressor complex, right? Because as we just went over, the repressor by itself is inactive and can't bind the operator anyway, but when bound to the co-repressor, tryptophan, it becomes active. So let's say that the operator can never bind this complex. So if it can never bind the complex, that means the transcription will never be blocked. So transcription never be blocked, and therefore the operon will always be on, which again is choice 3. Okay, part D. A mutant in which the TRPB gene introduces a stop codon in the mRNA. So remember that the TRP operon has 5 structural genes, A, B, C, D, and E. So it's saying that in the TRPB gene, it has a stop codon in the mRNA. Now, although in a polycistronic mRNA, each gene has its own start and stop codons, but it's saying that this one introduces a new stop codon. So basically, you'll get non-functional product of the TRPB gene. Now, each of these 5 structural genes either form a complete protein or a subunit of a protein for the biosynthetic pathway of tryptophan. So if you're making a non-functional TRPB product, that means that the biosynthetic pathway for tryptophan won't work. Okay? So this means the cell will be incapable of making tryptophan. Okay, so remember, biosynthetic pathway, it's like a stepwise thing. If you don't have the enzyme to go from step 2 to 3, that means you can't get to step 5 at the end, which would be tryptophan. So the cell cannot make tryptophan, which means that they will, unless it's introduced from the environment, which they didn't tell us to consider, if there's no tryptophan being made, that means that there's no tryptophan binding to the repressor protein, which means that the operon will always be on. Which would be number 3 again. Now, even though it's always on, because there's no levels to regulate it to repress it, it doesn't mean that it will ever make tryptophan, because as you see, there's a non-functional product within the structural genes. So the operon will always be on, but tryptophan will never be made. And then finally, part E, a mutant in which the operator permanently binds the repressor protein complex. So this operon will be under regular control. So under normal conditions, it will be on until an excess of tryptophan accumulates. Now at that point, so we'll say normal control until tryptophan levels are high. And when the levels become high, the excess tryptophan will bind to the repressor protein, make a complex, an active complex, which will bind to the operator. And in this case, it's saying that the operator permanently binds this repressor protein. So this means that transcription of the rest of the operon will be blocked permanently. So once it is repressed, there's no way for it to be unrepressed again. Okay, so permanent binding, it will never come off, and the operon will be completely turned off, which is number 4 phenotype. Okay, so this, again, we were just focusing on the repressor part, not the attenuation. There are 2 levels of regulation for the TRP operon, but this, I think, is a good, gives you a good overview of how the different elements in the regulation part of the TRP operon work. Problem 4. Attenuation is one example of how events occurring inside a cell control gene expression. Determine if the following statements are true or false as they relate to tryptophan attenuation. A. The untranslated leader sequence of the TRP operon is located 3' to the structural genes. So this is a small detail, but it's actually false. And that's because it says 3'. So remember that the leader sequence, which is the one involved in attenuation, is upstream to the structural genes, which means it is 5'. So remember, as I drew a few problems ago, the leader sequence goes 5', and then it's the different domains. So it starts 5', it's upstream of the structural genes. B. When cellular tryptophan levels are high, a stem loop structure forms between regions 2 and 3 in the TRPL mRNA. So again, this is false. Okay, so this would be true if it said that TRP levels are low, because remember, when tryptophan levels are low, the ribosome stalls on domain 1 because it lacks the tRNA for tryptophan, and therefore regions 2 and 3 can form a hair loop structure. But when levels are high, so when TRP levels are high, this structure actually forms between 3 and 4. And that blocks transcription. Okay. C. Control through attenuation works by turning off the TRP operon when the cell does not need the amino acid. So that's completely true. When you have enough tryptophan, the attenuation senses that, and it forms the 3,4 stem loop, which blocks transcription. So you don't need to make anything further, and you don't need to waste energy. D. The TRP structural genes will be transcribed when there is a stem loop structure between regions 3 and 4. So remember, as you just did in part B, a stem loop structure between 3 and 4 is what happens when tryptophan levels are high. And when that happens, the stem loop in 3 and 4 blocks transcription and turns that operon off. So this is going to be false. You can either change it by saying the TRP operon, this TRP structural genes will not be transcribed when there's a stem loop, or you can say the structural genes will be transcribed, but that is when the stem loop is between 2 and 3. Okay? So when there are high tryptophan levels, domain 2 gets kind of wrapped up in the ribosome because region 1 is translated so quickly, and therefore 3 pairs with 4, and the loop acts to stop transcription. Okay? So it's just basically saying to the cell, why waste your energy making more tryptophan when you already have it? Okay. And finally, E. Region 4 of the TRP leader sequence is considered the attenuator. Now, this is true. Okay, so it really depends on what textbook you're reading. Some say that region 4 is the attenuator. Others say that the attenuator is located more closely to region 4 than any other region. But the way I remember it is just that when 4 is stem loop base paired with region 3, that is when attenuation occurs, when transcription stops, okay? But when 3 is with 2, transcription occurs, so that's how I just remember that region 4 is considered the attenuator. Problem 5. The process of attenuation can help prokaryotic cells conserve energy, but would not be possible in eukaryotic cells. Based on the differences between these two cell types, explain why. So remember, as we've just been going over, that attenuation relies on this leader sequence, which has 4 regions, being transcribed and then soon after translated. So as it's being translated, region 1, which has a few tryptophan levels, if the ribosome stalls, then the stem loop structure forms that makes transcription continue, either between, if it stalls, it's 2 and 3. However, if it doesn't stall, if translation occurs very quickly, then a stem loop forms between 3 and 4, and transcription stops. So that's how they use translation to really sense how much tryptophan is in a cell, and therefore turn the operon on or off. So in a prokaryotic cell, this works fine. And that's because there is no nucleus. Now, because there's no nucleus, that means that transcription of DNA into mRNA and translation from mRNA into amino acids, proteins, can occur in the same space, right? Because there's no separation. There's no membranous organelles that separate out. So transcription and translation happen in the same place. And that's important because that means that translation can start before transcription is finished. And that's the most important key here. Okay, so let me star that out. And I'll just show you, if you have a piece of DNA, which we could show in blue, and you have RNA polymerase, I'll show that in green, and that's coming over, it's going down the DNA, and it's making an mRNA copy of the DNA. Now, because transcription and translation happen in the same space in prokaryotic cells, that means that a ribosome can come over as mRNA is being formed and can start translating it. So this is the ribosome, and then it's going to start spewing out the protein. Okay, so I'll just draw that as little circles for the amino acid. So before transcription of the DNA is even finished, ribosomes can come over and start translating it into proteins. So this is what's so important, because if this didn't happen, if the ribosome didn't come over until the entire DNA section was transcribed, then it wouldn't matter if the ribosome stalled on region 1 or not, right? Because the whole thing would be transcribed anyway. So it would eventually be translated. So because this can occur at sort of the same time, one right after the other, the activity of the ribosome, whether it stalls or not, can really change if the rest of the DNA molecule is translated. Now, in contrast, in eukaryotic cells, there is a nucleus, obviously, and this makes it so that transcription and translation occur in different places. So transcription occurs in the nucleus, and translation occurs out in the cytosol. Okay, so it's separated by space and time. So because the mRNA, we'll call this the nucleus. This is where DNA to mRNA happens. Then the entire mRNA has to come out into the cytoplasm. and this is where mRNA to protein occur in the cytoplasm. Okay, so as I said before, in this case now, the entire mRNA has to be transcribed before it even begins to be translated, because it has to be transported out into a separate space in the cell. So here, the activity of the ribosome, whether it stalls or not on the leader sequence, won't have any effect because the entire mRNA is already transcribed. So in attenuation, this really works by stopping transcription. If the ribosome doesn't stall, then it stops transcription and the entire mRNA isn't transcribed. And this is really why. So it's really because of the separation of different cell compartments, the compartmentalization of it with a nuclear membrane is really the difference, and why attenuation wouldn't be possible in a eukaryotic cell.